let's begin. Before we jump into meiosis, we need to refresh ourselves kind of again on what mitosis is, what we expect from normal cell division before we jump on to meiosis. So, like I said, cell theory says that all living things are made up of cells, including the isopods that I just showed you. Cells are the basic unit of life, and all cells come from pre-existing cells. This is what we're talking about right now, that all cells come from pre-existing cells. So, we start out in our interphase. This is on your first section of notes. In interphase, we have those three phases. We have the G1, the first growth stage, when the organelles are going to duplicate. The S phase down here, where DNA replication, the thing we talked about on Monday, takes place. So S is actually going to stand for synthesis. Synthesis is to create. So this is where we're going to be creating our new copies of DNA, kind of like you did with the connects. And then last but not least, we have G2, which is going to be last-minute preparation so that we can go into mitosis. So this is going to be 90% of the cell's life. Most of their time is just going to be spent growing, eating, regulating themselves before they jump into mitosis. So the other thing we need to refresh our memories on, like I said, we've been doing it over the past couple days, but we need to know the difference between somatic cells and gamete or germ cells. So we are going to be moving away from somatic cells. That's what we've been talking about. Matthew, what are somatic cells?
Oh, great.
On the board. They are diploid. They are our body cells. So all of your skin cells, all of your muscle cells are all going to be somatic cells. They are diploid, so they have the proper amounts of DNA for your organism. In this case, we have 46 chromosomes, and they're going to reproduce through mitosis. So that's what we've been talking about. Now we're going to move on to gamete cells, or germ cells. And these are going to be your sperm and egg, your sex cells. They are going to be haploid. So haploid is half. They have half of their chromosomes, and they reproduce through meiosis. So that's what we're going to be talking about today. Except this should all be review, just kind of getting it into your head, all of this part, before we move on to meiosis, because a lot of this is going to be very similar to things you've already seen. It's just going to be arranged differently. Anyone need more time? The other thing we need to talk about is our nuclear material, so our DNA. So in both phases, whether it be mitosis or meiosis, we are going to have a division of the nucleus and the genetic material within it. So whenever we move into things like mitosis and meiosis, we are going to condense our DNA down into chromosomes. That way it can be easily moved. Think of it like a ball of yarn. Is it easier for me to carry it around like this or to carry it around like this? The first one. If we roll it up into a ball, it's very easy to store. It's very easy to transport. Same thing's going to happen with our chromosomes. It's much easier to move it around as a chromosome than it is as loosely organized chromatin. So chromatin is our unwound double helix, very stretched out, very loosely organized in our double helix, like I said. And this is going to be found, chromatin is going to be found in the nucleus during interphase. So typically whenever we look at a cell that is not going through mitosis, it will have a very dark nucleus because it's just filled with chromatin. There's so much chromatin in there that it's actually just a dark circle instead of the little squiggles like we've been drawing. And it's also good to note that in each cell there's about 1 meter of DNA. So for each cell you have about this much DNA in each one of your little cells. So all of it has to be wound up very, 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 very tightly in order to pack it into our cells. And we still need to talk about, like I said, our haploid and diploid. Haploid has half of the total amount of chromosomes. Diploid has the same amount. So di is 2, so usually there's 2 copies in each chromosome. So they're going to have the same amount of material. So for our cells, we have 46. For our gametes, we have 23, yes.
So for the study out, was that graded on completion?
Yes.
Like the effort instead of the reaction?
You have to get over an 80%.
Okay, and then just continuing on with review, we have our chromosomes. What are the little green things here, Ashlyn?
The green thing is
Yes.
the centromere.
The centromere. And we have our chromatids. 2 chromatids together form a chromosome. So they are sister chromatids. Like I said in our class demo, each of you in your collaborative pairs were sister chromatids. These chromatids come together to form chromosomes. However, the one I mentioned before and did not cover too much, homologous chromosomes, we are going to be talking about a lot more today. So homologous chromosomes are going to be very important with meiosis because we're going to do some stuff with them that we did not do in mitosis. And last but not least, can someone tell me the stages of mitosis? How we go?
No, oh, I was trying to think. It's PMAT.
Okay.
You get
So PMAT, PMAT,
prophase,
prophase,
metaphase,
metaphase,
anaphase,
telophase,
cytophase.
and cytokinesis. Good.
I
Yes.
couldn't remember.
So we have our interphase, our normal cell life, prophase, nucleus breaks down, chromosomes start to form, metaphase, chromosomes line up at the middle of the cell, anaphase, we start to have disjunction, we start to have the cells or the chromosomes pulling away from each other. Telophase, we have the nuclei coming back together to form 2 new nuclei, and cytokinesis, finally the cytoplasm pulls away, and we are left with 2 new cells. So in real life, if I were to throw a couple slides under a microscope, this is what mitosis looks like under the microscope. So we have our first one. This is interphase. You can see that the nucleus is very dark, very filled in. It's full of chromatin, so you're not going to be able to see through it. But when we start to enter mitosis with prophase, you can see that it's all starting to kind of clot up and form those chromosomes. In metaphase, we see that they are lined up in the middle. Anaphase, they are starting to pull away to the different poles, the different sides of the cell. Telophase, we start to see a new nuclei coming back together. And finally, cytokinesis, they are fully separated. So, I hope these 4 videos don't go off at the same time. So what we're going to do is I'm going to play a song. Within that time, you need to come up with the 4 main points of mitosis. Any questions? Okay. So we have, in terms of key points, we have PMAT, so prophase, metaphase, anaphase, telophase, 2 identical daughter cells. What else? Ashlyn.
Diploid cells reproduce through meiosis.
Okay, so it's going to make diploid cells. And what's one more? Abigail.
Interphase is like the intro and cytokinesis is like the conclusion.
Okay, so we start with interphase and with cytokinesis. Yes. So, just looking through here at my notes. Anything else? So what happens to our chromosomes during mitosis?
They
They split apart. Yes. So what stage is that? It's an anaphase. Yeah, so it's going to divide at anaphase. The chromosome is going to be split into its sister chromatids. Any questions about this? So keep this in mind because meiosis is going to have some of this that is very similar, some of it that is very different. So, before we go into actual meiosis, I want to just cover a little bit of genetic variation. So, it's not on there. Up there it is. So, on your paper, there's a spot for the activating question. I have 2 questions for you. Think about these independently and jot down what you think. Do you look like your parents? Why or why not? Jot it down. And do you look like anyone else in the room?
No.
Why or why not? So write it down. Okay. Raise your hands. How many of us think that we look like our parents?
We
Okay.
don't.
So do. How many think we don't that you don't look like your parents? Okay. Okay. So Brayden.
Because you have like
What is it?
genes and genetics and stuff from them.
Okay.
It's half of your mom's and half of your dad's, I guess.
So your genes. So 1/2 comes from your mom, and 1/2 comes from your dad. Now, how many of you think that you look like someone else in the room?
I don't look like Owen. I don't think I look like me and Owen are actually twins.
Uh-huh.
Separated at birth.
Uh-huh. Okay, how many of you think you don't look like anyone else in the room?
I don't know.
Okay. So, Emery, why do you think you look different from everyone else in the room?
Crazy. That's crazy.
Okay, so you have different. So Emery's genes may look different. The different alleles, different things like that, may look different from Matthew's, or may look different from Owen's, or may look different from Kyle's. So everyone has different genes that are passed down, and they are going to look different. But we're going to look into more of the biological aspect of why this happens, or how this happens, so that later we can talk about why it happens. So in terms of genetic variation is only going to happen in meiosis. And there's a couple ways we do this. The first is called crossing over. So this is a process during meiosis where we have our chromosomes, and they're going to, for a little bit, hold hands. So they're going to do what's called synapsis, and they're going to wrap one of their arms around the other chromosome. It's like giving them a hug.
That's weird.
It gets weirder. So whenever they do this, crossing over, actually before I said this, so this is only going to happen with homologous pairs of chromosomes. What do I mean by homologous chromosomes?
Weird.
No, what do I mean by homologous chromosomes? The same. So we have 23 pairs of chromosomes, 22 autosomes, and 1 pair of sex cells, or 1 pair of sex chromosomes. So in this case, chromosome 1 is going to link up with chromosome 1. Chromosome 20 is going to link up with chromosome 20. So they are going to link up in what's called a tetrad. Tetra is 4. So 4 chromosomes. So they're going to start with synapsis. They're going to wrap around each other. And then they are going to cross over. This crossing over is when they pull apart and swap genes from one chromosome to the other. So they are literally going to share parts. So for example, we have a red and a blue. After crossing over, one is going to have the blue will have one of the red side, and the red will have one of the blue side. Whenever these are separated, it is called disjunction. So this is the first way. So this is literally going to swap genes from one chromosome to another. So Peyton, if we were to separate these out, meiosis, we said, comes out with 4 cells. How are these going to be different? No. So we have 4 cells. Let's say 1, 2, 3, 4 cells. So each chromatid is going to make come into its own cell. How is chromosome 1 going to be different from chromosome 2? Well, what do you notice? Okay, it has part of red. Does the first one have part of red? So where did the red come from? The other chromosome. So this one, let's say chromatid number 1, is purely dad's side. Chromosome 2 is mostly dad, but we have the genes that we swapped from the mom side. Chromosome 3 is going to be mostly mom, but a little bit that we swapped from dad. And then chromosome 4 is going to be purely mom. So that is how we're going to vary a lot of the genetics in this case. We also have the law of segregation. So this law states that for each gamete that is produced, it is only going to have one copy of a gene. So if we have a gene, let's say these 4 chromatids are all coding for the same gene, each chromosome or each chromatid there's only going to be 1 per. So this is going to help us out when we talk about fertilization, because we only need 1 copy from mom, 1 copy from dad to get our diploid pair. So this is going to ensure as much genetic diversity as possible. Yes, which is good, because if we don't, then we're going to have all sorts of issues, which we'll talk about those probably Monday is my guess. Any questions about this? Okay. So now we are going to go into actual meiosis. So you have a big chart, kind of like we did with the first one. So on this, you don't have to draw them, but I want you to at least jot down what's happening in each stage. So as you can see, we have not 1, but 2 phases this time. Yes. We have meiosis 1 and we have meiosis 2, but you'll notice it's the same steps that we've been using. So our first stage is prophase 1. So we note them as either 1 or 2. In prophase 2, we are going to, just like in prophase of mitosis, we are going to condense our chromatin into chromosomes. We are going to break down the nuclear envelope. But in prophase 1, we're going to get crossing over. So we have that tetrad, those homologous pairs, and they're going to swap their genetic information at this point. So as you can see in the diagram, they're kind of hugging together, and they are going to actively swap their DNA. So this is diversity checkpoint number 1.
We're making cookies.
So we then move on to metaphase 1. This one's going to act pretty much the same as our metaphase in mitosis. The only difference, as you notice, we have our homologous pairs. We have 2 chromosomes. We are going to line up, again, in the middle, with 1 set of chromosomes on one side, 1 set on the other side. So we're not swapping chromatids at this point. We are swapping whole pairs of chromosomes. So that is the main difference to look at in these first steps, is that we are not working with individual chromatids. We are working with whole chromosomes. But otherwise, this is going to look the same. So we have our centrosome up top. It's going to grow out its spindle fibers and connect to all of the chromosomes.
Why is that biology? He's amending. That is
Our next phase is then anaphase. So we're going into anaphase 1, guys. So anaphase 1, like I said, instead of pulling individual chromatids apart, one chromosome is going to one side, one chromosome is going to the other side. And this is going to be this is going to be disjunction. So disjunction is where we have the separation of the homologous pairs. So your dad's side chromosome is separating from your mom's side chromosome.
the equator.
Yes. That is the middle of the cell, the equator. Kind of like when we look at the Earth has an equator. Same thing with these kind of equators. However, the equator in this case is a lot less noticeable than on the Earth.
Is segregation and then dads? Oh, it's what's the word? This is triggering to some of the audience.
What?
It's triggering.
You notice I'm using a biology context though. So after anaphase, what do you think comes next?
Telekinesis.
Telophase. Telophase. So telophase and then cytokinesis is going to happen. Yes. So in telophase, we are going to create two new nuclei, but in this case, we are going to create two new nuclei full of chromosomes. So we have half of our chromosomes on this side, half of our chromosomes on this side. And then, of course, cytokinesis, the cell or the cytoplasm is going to divide. And this is going to now result in two haploid cells. So we are no longer diploid, we are haploid. So haploid means half. So we have half of our DNA right here, half of our DNA right here. So like I said, mom's side goes to one side, dad's side goes to the other. So we are going to split it in half. Now we have 23 chromosomes on this side and 23 chromosomes on this side. Yes, Peyton?
Start.
With what?
Oh, nothing. I'm just writing down notes. Okay, wait. Yay! Wait, are we just ending on the telekinesis like stuff? Are we
No.
doing this whole pack?
There's a couple other things. So, you notice at this point we have hit the point where normally in mitosis we would stop. Mitosis or meiosis doesn't stop. Now we move on to stage 2. And yeah, so like I said, what does it look like? Looks like the stages of mitosis has just been using chromosomes. Now it's going to look like mitosis. So in prophase 2, again, we are going to create new spindle fibers, and we are going to go and dissolve our nuclei.
I don't have anything.
Can you just think about Can I utilize it?
So prophase 1, going to look like the same thing we saw in mitosis.
It's a 2% job. Girl, simply. Oh my word.
Then we move on to metaphase 2. Metaphase 2, again, just looks like mitosis. We have our chromosomes lined up in the middle of our cell at the equator. However, you notice we're doing this twice as well. So in one cell we're doing this, and in the other cell we're doing this. They're going to line up in the middle at the equator. Like I said, if you think equator, think of the cell as a planet. We have the equator that runs around the middle of the cell. We have the poles that sit on the top and the bottom of the cell. Then we move on to anaphase 2, which again just looks like mitosis. We're separating the individual chromatids now into each cell. So the centromeres are going to divide, and the chromatids are going to move towards one end of the pole or the other.
They should be brought to the front of the room.
About ready to.
Oh, snap.
His glasses are so pretty.
And last but not least, we have our telophase 2 and our cytokinesis.
Yay.
We form 2 new nuclei, each one with one set of chromosomes. So we end up here with 4 haploid cells. So they all have half. One half is in this cell, one half is in this cell. So you see in mitosis, we did prophase, metaphase, anaphase, telophase. In meiosis, we did that twice. First time we separate homologous pairs of chromosomes. Second time we separate homologous chromatids from each other.
Oh, Ashley.
I'm gonna have to cut that one out.
What?
Okay, so I'm going to play a little bit of this video. I'm not going to do the whole thing. Hopefully it works because it's very old. So, guys, focus. So we are going to create, by doing crossing over, what are we going to create, Peyton? By doing crossing over, what are we going to create?
Variation.
Variation.
We're swapping genes.
How much variation?
A lot.
Okay.
PDA. Oh, what?
Oh.
Okay. Kyle, what's another thing about meiosis?
Each species ends up with one copy of each.
Okay, so what is that called? No, what is the cell called? If it has one copy, as opposed to normally we have 2, we have haploids. We are going to create 4 haploid cells.
Like here?
So we're going to create 4 haploid cells. Matthew, what happens in meiosis 1?
Prophase 1, metaphase 1, telophase 1.
Okay, what are we separating?
The nucleus.
Thank you.
What version of DNA?
The homologous chromosomes.
So we are separating chromosomes in meiosis 1.
Oops.
So in meiosis 1, we are specifically separating pairs of chromosomes. Noah, what happens in meiosis 2? What are we separating?
I think you're separating.
Look at the notes.
Chromosomes.
Into
You said metaphase, right?
Into nuclei.
no.
That's what you said. Why do we need to know that?
Why do we need to know that? So you know, so you can tell the difference between the two. So,
Skip like that, Carter. I know you do.
what is half of a chromosome called?
Chromatid.
Chromatid.
Yeah.
You're separating chromosomes into. If you do not have these two parts, you will need to get them done because those are going to be very important. That's the main way that you can tell if something is meiosis 1. Meiosis 2 is whether we are separating chromosomes, so we're separating the X's, the pairs of chromosomes, or if we are separating chromosomes into chromatids. That is the main way you can tell. Otherwise, all the steps look the same. Okay. Another minute, finish up. Anyone need more time? If not, we're going to jump to the very fun part. Well, quote unquote fun part. So, you can see here, mitosis, we went from one diploid cell to two diploid cells. Each one has a copy of both of those. In this one, we had meiosis. We have our pairs, our tetrads, split apart to form two haploid cells. Those haploid cells then give each copy to the daughter cells to create four haploid cells. I'm going to skip this for now just so we have time. All right, going to jump to this part.
Oh boy. Oh, the very reproduction.
So, in meiosis, we have two different versions. We have spermatogenesis, not tomato. No. Spermatogenesis and oogenesis. So spermatogenesis is going to be the one that all of the males in the classroom are going to do. Oogenesis is what all of the females are going to be doing.
What?
Yes. So all of the males in the room are doing this one.
No, that's why I call it. You can't do that nowadays.
Relax. All right, focus. So, in spermatogenesis, we are going to be creating sperm cells. In oogenesis, we are going to be creating egg cells. Those are our two gametes. In spermatogenesis, we are going to end up creating 4 sperm cells. So each one is fully functional. Each sperm cell is haploid and unique. They all have their own different codes thanks to what two processes?
Spermatogenesis.
No. What helps create the variation? What in meiosis?
It's like the chromosomes like swapping.
What is that called?
Crossing over.
Crossing over and the law of segregation.
Oh.
So those two are going to create the variation that is needed to make all of these unique. Like I said, how many of you have siblings?
Sadly.
I know.
How many of you look exactly like your sibling? Identical to your sibling.
I'm adopted.
So in this case, crossing over is going to help kind of separate that. So you may not look exactly like your sibling. You may have some shared characteristics that came from your parents, but not all of them are going to be the same. And in this case, sperm is going to be produced throughout a male lifetime. So you will always have access to it. Sometimes it will be more viable than others, but you will always have access to it. In oogenesis, we are going to create one egg, which is weird because we said that in meiosis we created 4 cells. The other 3 are going to be produced as these polar bodies. These are little cells that more recently have been thought to nourish the egg. One of these cells will come out to be a proper fertile egg. The other 3 will pop out to be these polar bodies. Each egg, same as the sperm, is going to be haploid. And it's going to be unique in its genetic code. And all of you ladies in the class were born with all of the eggs that you will ever have, and you will not make more. So you have many, and you will not make more. It's you develop them throughout, I forget how long it is, and then you stop producing them. So all that you will ever need is what you have right now.
Yes, you do.
Yes. So, Cade, why do both of these need to be haploid? Why do you think?
They're both sex cells.
Okay, so what are they going to end up doing? They're going to combine. If we combine two haploid cells, what do we get? A diploid cell. So we need half of our genetic material in one cell, half of the genetic material in the other cell. That way we can bring them together and form a fully diploid fertilized egg. So in this case, one side came from your mom, one side came from your dad. Those two haploid cells combined to give you your diploid cells. Any questions about this? So then with this reproduction, we have two different forms. Anyone think they know what the two forms are? Asexual and sexual. Sexual reproduction, we are going to use mitosis. So that's on your last page. Sexual reproduction is going to require meiosis to create our gamete cells. We are going to combine our egg and sperm to create a fertilized diploid egg. One set of those chromosomes are coming from the mom, one of those chromosomes are coming from the dad. And each offspring is going to be unique. Anyone think they know why? Like I said, why are you different from your siblings?
Because we're different cells.
The genes are different. So we have these homologous chromosomes that they're going to cross over, and then they are going to be separated up into individual chromatids by the time we get to sperm and egg. So each one of your eggs, each one of your sperm cells are going to be completely different from the other ones. You will never, or in this case, you should never have two eggs or two sperm that come out with the same result, because that is going to help increase the genetic diversity.
What happens with twins?
So twins depends on, yes, that is a good question. Twins depends on what kind.
What a great.
Because sometimes it's just a matter that two eggs are deposited at the same time. In the case of identical twins, it's actually that the cells kind of come apart during replication. So we have one egg that is fertilized, and in this case it kind of breaks in half. So half of it develops into one person, half of it develops into the other. That's how you get identical twins. Fraternal twins, it's more so just two eggs are developing at the same time.
Stop trying to sound smart, please.
So let's finish this up. So, our last thing we were going to talk about and what's going to lead us into our activity for the rest of the period is asexual reproduction.
I know.
So, typically in nature, asexual reproduction is going to happen with mitosis, not meiosis. Why do we think it happens with mitosis?
Because asexual reproduction results in the exact same identical cells.
We are creating identical cells. We don't need to combine sperm and egg to create a fertilized cell. We only need to divide. So each offspring then is going to be diploid. It's going to get one copy from the parent. They're going to be identical. So in this case, yes, whatever the mom had in this binary fission is what both of these babies are going to have. So this is also going to cause a little bit of issues because there's less variability with asexual reproduction than there is because there is no crossing over. There's no crossing over, there's no law of segregation, so there is no way for these guys to get new genes in. That's why bacteria has to evolve over time to get new genes. So this can be done in a lab through meiosis. For example, we have been able to clone things like sheep and different animals, we have learned how to successfully clone them in the lab. I think the first successful clone was a sheep named Lucy, I think. Yes, we have successfully cloned things. And that was done through meiosis. We had to artificially combine eggs and sperm in the lab. No.
Wait, I have a question.
Yes.
So if you were to, was the whole thing working? If you were to try and clone somebody, they wouldn't come out as the same age that they are.
No, it would start from single cell and have to grow.
So would it grow exactly like that person?
So that's one thing we'll talk about in genetics is the kind of interaction between nature versus nurture. So whether it's your genetic code that determines what you're going to look and behave like versus your environment. So so that's when nature and nurture comes into play. Even if you raise something the exact same way, there have been twin studies that figure out that twins, if they are raised in the exact same conditions, don't even always come out the same. So there is a lot of variability in this. But a couple examples. Binary fission we've talked about. Budding we've kind of talked about. Fragmentation we've kind of talked about. Binary fission is just the splitting of a cell. Budding is something that hydras do. They create a little baby that pops off. Fragmentation, if you've ever broken off a starfish leg, it will regrow that leg, and then the leg will regrow into a new starfish.
Unless you soak them in vinegar, then they just die.
Yes, that's why the crown of thorns is invasive off the coast of Australia, because it's a starfish that reproduces by fragmentation, and the more you tried, or people thought that the best way to kill them was to cut them up, and then it just exploded the population.
That's cool. What idiots.
So the other one, and the one that we are going to be doing for the last couple minutes, is propagation. So how many of you guys like plants?
I don't like.
I mean, I like eating them.
Okay, how many of you like growing them?
Oh, bro, I have little chicken keys or chicken.
Hens and chicks?
Ah,
Yes, Echeveria.
yes.
Last but not least, any questions with this before we move on? So, what we are going to be doing is we are going to be learning about propagation. So plants are really cool because plants are essentially just a bunch of little nodes working together as a group. And what you can do with that is that you can grow new plants from pieces of old ones. I do this all the time. I actually have a propagation bin back with my carnivorous plants. I have grown many tropical plants just from cuttings. Yes. So what we're going to do is I'm going to have you break up into your lab groups. I'm going to bring around cups, soil, and I forgot my sphagnum moss, so it's fine. I'll bring around the cups and the soil. What we are going to do is you have 3 different options over here on the side of the room. We have a golden pothos, Epiprenum aureum. We have a Baltic blue pothos, so this is Epiprenum canatum Baltic blue. And we have, I believe this Echeveria Dark Night, I think is the name of it. We are going to be producing babies from these plants just by using leaves. Yes. So what you're going to do is, if you get the pothos, you will cut a 1-inch segment around a leaf node. A leaf node is where the leaf is coming out of the stem. From there, you will stick it in a cup of damp soil, and then we'll put them in a tray and put them in a growing chamber so that they can successfully root. Echeveria, the hens and chicks, you'll cut off a single leaf. I don't have as many of those. I'm not going to do as many of them. But we'll pop off a single leaf, stick it in soil, and it will grow a new plant. Any questions about this? Okay, so we have a little bit of time, so we're going to try and rush this. If we don't, we may start it on Monday.
Okay.
Break up into your groups.
I'll frame it.